with the phosphate. So if we have this OO there, it can do that. Remember, that was in our enzyme worksheet. And then we would break this bond. We would catalyze that reaction to break it down. That's how you break down RNA or DNA. Okay? So we need to remove that. A binds to T in DNA. A binds to U in RNA. Very similar structures. Okay? Uracil and thymidine are very similar. We just have an extra methyl group right there. We bond on that nitrogen with a covalent to carbon 1. This way it's not shown. So that binds off there. So what are the differences and similarities of those two? What do we call it?
Purine.
Purines are which ones?
The big.
Big rings, small word. Big rings, small word. Those are big rings. Small word, which is purines. Pyrimidines, big word, small rings. Pyrimidine must bind to a purine. They bind together using what type of bond?
H bond?
H bonds. Excellent. H bonds are relatively weak. So how easy is it to unzip DNA? Pretty easy. Do you want it to be tough to unzip DNA? No. Because what happens when you unzip DNA? You're able to replicate it. You're able to transcribe it. Right? You're able to do things from it. You need to be able to access that information. So if I want to make it more easily accessible, what will be my sequence? More accessible. So I need to make it weaker. What will be the sequence? Try to figure that out. More accessible. So what will be the sequence of DNA?
I don't know, I'm thinking about the central dogma, so I'm thinking DNA, RNA, protein, but I don't know about that.
You want it to be more accessible, so what will be the sequence? A and T's. Excellent.
Oh, okay.
ATA, A and T's. If you have a whole bunch of repeating A's and T's, now you have only 2 bonds holding each nitrogenous base together. If you want to make something less accessible, you will fill it with what sequence? G's and C's. This is your first step to regulation. That's it. If I want to make something really accessible to being transcribed, I make it in A's and T's. We call this our TATA box. TATA means just repeating T's and A's. Whole bunch of T's and A's together. A protein comes along and says, whoa, shit, that's really easy to break apart. Let's break apart there. And we transcribe from that location. So if we have a whole bunch of G's and C's together, it's going to be harder to break. In fact, it's 1/3 harder to break. Right? Because now you have, or it's actually, no, it's 50% harder to break. Because of your math.
GCs. But
That's part of regulation. We'll get there.
Which protein?
oh, well, what do we do?
Each portion
Ooh. Could be that one though.
All right, Dr. Shark.
Oops. I was between that and that, but now I think it's adenine.
Yeah, I was between the sugar
Me
and
too.
adenine.
What do you think?
should grab one.
Huh?
I should grab
Hydrophilic or phobic?
Hydrophilic.
Very philic. So this is your phosphate backbone, really philic, negatively charged. Philic or phobic?
Philic.
Very philic. Sugar, right? We're gonna easily philic. Philic or phobic? Phobic. They can form 3 hydrogen bonds. So that's relatively phobic.
And because, are the ends locked there?
Everything is, yes.
Oh. Oh, why did I put this? Really? Jeez. Wait. Okay, finally.
Alright, talk to each other.
F.
I said C.
Hydroxyl. Oh, it is hydroxyl. You are right. You're right. I was thinking DNA.
What's the only difference? Hydroxyl group is now gone on carbon 2, therefore it is 2.
Wait.
They're in different order up there.
Oh, okay. Like, so.
To recap, DNA is most of the time double-stranded. It goes from 5' to 3'. That's how we read it. We build on the 3' side, so DNA is elongated on the 3' side. DNA does not have a hydroxyl on carbon 2. DNA has a proton. RNA has a hydroxyl on carbon 2. RNA also builds on the 3' side. We always build on the 3' side. The difference is RNA, for the most part, is single-stranded. For the most part. The bases we use, T, C, A, and G, we replace T with U. That is it. Okay. How we feel? Next. Draw those hydrogen bonds.
Oh. I better look over here because they're quite different. That would be GC. GNC. Shoot.
About 10 seconds. All right, talk to Trevor.
I put B.
GNC3 and ANT2.
Yeah. Because the
Yeah,
nitrogen
you are
right on the A can go to the
A and T's can form how many hydrogen bonds?
2.
2. G's and C's can form 3. Is the hydrogen bonding here?
No.
Where on A and T's is it? Is it here?
No.
No. Why isn't it not here?
The hydrogen is connected to a carbon.
The hydrogen's connected to the carbon. That is covalently locked. That cannot do anything.
Forgot about that.
Our hydrogen bonding is there and there.
Yeah.
There's our A and T's.
Mm-hmm.
Right there and there. On the G and C, I can go here, here, and there. Therefore, I have 3 of them. Okay?
Got it.
If I have 8% of my DNA strand is G, how much is C?
8%.
8%. Okay, Dr.
I have no idea.
I said 20%. Because if 30% is guanine, another 30 has to be C.
If 30% is G, therefore how much is C? 30%, therefore 60% is now gone, right? Because we have G equals C. So then you need 40% divided by 2 is equal to 20%. So G to 3, A's and T's do 2 bonds. All right, so that's our orientation within the nucleotides. So then this is carbon going to bind to carbon 1. Okay, so that's carbon 1. That will be our directionality. Okay, then this is going to bind to carbon 1. So that will be our directionality. So this side must be pointing, 3' must be pointing up. This side must be 5'. As anti-parallel. Which carbon?
1.
1, 2, 3. 3' is pointing this direction. 5' is pointing
That way.
that direction. Carbon 1 has the nitrogen space on it. Which carbon is that?
1, 3, 4, 5.
5 is pointing this way, 3' is pointing that way. So if 3' is pointing that way, 5' must be pointing that way over there, anti-parallel. Okay? So when they were first making this discovery, we knew the chemical components of this. We knew that it was made out of phosphate. We knew it was made out of sugar. We knew it was made of nucleic acid. We knew all of these things. We, in fact, knew the quantities of them. We knew the A's and T's had to be equal to each other, so they had to be binding. We did not know the structure of it until there was a paper. And so we have Watson and Crick and Rosalind Franklin. And so we had all of these repeating distances. And what was so important about this is that Rosalind Franklin was a biochemist. Watson and Crick were not. They were organic chemists. And whatever they did, they knew the structure. They knew how it worked. They couldn't figure out how to do it. And they couldn't figure out the actual three-dimensional structure. And that was because every time they were trying to do X-ray chromatography, they would put it in some sort of crystalline structure. They'd put it in an organic solvent because that's everything they knew. Well, what did Rosalind Franklin do? Put it in water. When she put it in water, it formed this beautiful helice. And forever, people have been saying that she was kind of screwed, that she wasn't given the credit she was deserved. And so Watson and Crick published their paper, And they did no experiments themselves. What they did was all the computational, and they did the modeling. And they used lots of other people's empirical evidence, and Rosalind Franklin's is what they used to confirm it. Watson and Crick are mixed people historically. A little bit racist, a little bit, on the side of eugenics. So, we'll good and bad. But there was this whole idea that Rosalind Franklin, you guys have probably been taught this, that she was the victim, the heroine. She was that. In fact, history is kind of rewriting itself. It is thought that Rosalind Franklin was not the victim today. So it's not a good example when we talk about women's suffrage and women being exploited in STEM. Do not think I am saying women aren't exploited in STEM. We'll go into that here in a second. But a lot of the work done today actually suggests that from her perspective, she was very well thought of and was given credit. The problem is she died really young. And when you die really young, sometimes your work goes unnoticed or it gets picked up by other people. And that appears to be what have happened. So she is not just some random footnote, and she really looked at this. She probably deserves to not be considered to be wronged by science, because she was very much considered an equal in their lab. The correspondence between them was very much equal. There was a lot of invitings to events, things like that. So that's something to think about. So when we think about history, it's something to always remind ourselves. With that said, being a woman in science does suck. Often it's not as glorified. We have talked about why there are so many more men as PhD holders in the past. I was doing an advising over just a few days ago, and an older student wants to pursue it. They're coming back to science, and the likelihood of them being a PI is very, very low, just because of their age. There's discriminatory practices. It's our society. But at the same time, fight for yourself, always stand up. I know quite a few labs who, if you do that work in that lab, the PI takes credit for it, even if you are the one who did it. So you're under their umbrella, they'll take the credit. My lab, we try not to do that. We have undergraduates on our papers and graduate students. That's more the new norm, but not always the norm. Anyways, so this is just a great example of history of science, but also understanding your world. Rosalind Franklin just did this simple experiment of throwing it in water, doing the chromatography. Don't do it as crystalline chromatography. So, thought that was interesting. All right. So, what she knew was the structure. What Watson and Crick knew was the structure. They didn't know anything how it worked. They didn't know the enzymes involved. They didn't know how the reaction is polymerized. They didn't know how it catalyzed. We now know all of that. So what we're going to do is we're going to take a charged nucleic acid. Charged nucleic acid just means we put a bunch of phosphate groups on them. Those phosphate groups have that tilde, which represents what? High energy bond, right? Strong bond. So we're going to use that high energy bond to catalyze this reaction. And the reason why we can do it on the 3' end is we have these phosphates attached to it. I don't have phosphates attached here, so I can't build on the 5' side. So I'm going to take that, we're going to catalyze it, we're going to knock those off. ADP is going to steal them. We're going to get some, have some inorganic phosphate floating around, and we're going to create that ester bond, that ester linkage. And so we're going to continuously build on the 3' side. Well, you'll notice how it's stair-stepped. And when you have a stair step with the hydrophobic inside, that's what's going to form that helice. And it's going to come back down and around. And so this week in lab, by the end of lab, you will have to know how to construct a helice. And we'll be doing it so you can see how the bonds work. What did I just say? memory of what I just said is not so great.
What did you do?
I did B.
I did too.
I did A.
Maybe you're right. Who knows?
So is it A?
Two of these I said. One I said, but in reverse. Inorganic phosphate is not attached to anything. It's released into the environment. A is incorrect. The other two are correct.
Oh, I was between the two bottom ones. Okay, not bad.
What end of DNA is here?
3 prime?
3 prime. What end of DNA is here?
5.
How many bonds?
2, 3.
3.
What type is this?
Guanine?
Guanine.
Purine.
Big ring.
Appearing.
Purine.
Small word. Purine. This is a
Pyrimidine.
Primitive.
go.
Oh, so what?
Most DNA that you would ever observe is going to be in a condensed form. Condensed form is what we had when we did this little thing. Mom, Dad, and they replicate.
Replicate.
What are we doing this week?
Replicating.
Replicating. We're learning this step, okay? So condensed DNA is how it looks in the real world. DNA, if you were to stretch out of your cell, single cell, we're talking, it's really long. Okay, it's very, very long. So what we're going to do is we're going to wrap it around something that's sticky. Well, the outside of DNA is what charge? Negatively charged. So if I put something that's positively charged, what can I do to it? Ionic bonding, I can bond it together. So I'm going to put histones. So I'm going to histonate it. All right, so we're going to histonate it by putting histones. These green things represent histones. Those histones allow DNA to coil around it. Basically takes a little ball and we're going to ball DNA up. Now, this allows it to be condensed down so you can move it around, you can do things. But also, do you need access to every gene in every cell? No. So, if you have a muscle cell, you need access to your muscle genes. Do you need access to your cardiovascular genes? So what can I do to it? You can shut it off. How can I shut it off? What if I just wrap it up and put it into a ball? That's what it works. We're going to histonate it. Okay? If you histonate a gene, you wrap it up. Now, can you still express that gene occasionally? Yes. But is it going to be more likely or less likely?
Less likely.
We're going to histonate it based off the polarity of it. Okay? You have a ton of DNA, all right? You have 23 pairs of chromosomes, as we've talked about, 23 in me, right? You're 2N. 2N equals
46.
46. So you have 23 pairs. You are a diploid organism. You actually have 27 chromosomes. You have 23 linear chromosomes, and you have 1 circular chromosome. Circular chromosomes are what bacteria have. Okay? Why do I say you have 47 chromosomes? What circular chromosome would you have? Talk to her. Try to figure that out. What circular chromosome do you have?
The X or Y, I don't know. X or Y. I don't know what to say.
You already know. We did this already like 2.5 months ago. We did. We're going to work on that.
Meiosis or
What is the extra chromosome?
A circular chromosome.
It's a circular chromosome. Who is it? It's mitochondrial chromosome. You've heard of mitochondrial DNA. Well, mitochondrion is just a bacterium. It has its own DNA. It has its own circular chromosome. Mitochondrial chromosomes, like bacteria, are a circle. Means if the gene starts here, it's going to go in a certain direction. That's it. You can have your polarity based on that. So if it starts here, it goes there. If it starts here, it can go there. I don't know. They can overlap. Genes can overlap. Genes can be expressed in multiple places at the same time by multiple things. Many genes are duplicated in chromosomes. So there's a lot of nuances to this. So if I say you have a gene on 23, right there, that pair, that would be your sex chromosomes, X and Y. That's why they look so different. All the other are homologous. So mom, dad replicate, right? So these are sister homologs. These are sister chroma. There we go. We're remembering things. Let's practice that.
You said we had 23 and then 1, but then you mentioned 27, or did I hear wrong?
You
What?
missed 47 total chromosomes.
Oh, 47, that's what it is. Okay, got it. Thank you.
Okay. DNA typically comes in a form of double helix, double-stranded, double helix. RNA is typically linear. However, nothing wants an exposed end, okay? In chemistry, everything wraps up on itself. So it'll wrap up and the nucleotides will start bonding together. So it becomes a classic combination sequence. This is called a hairpin. Hairpin loops are just where the RNA has wrapped up on each other. It can form many different things. It can form things like a ribosome. It can form tRNAs. That's your tRNA. So a tRNA is an RNA sequence. Okay? It's wrapped up, it's formed as helices. It's forming tertiary structure and quaternary structure. And this anticodon here, which is a 3 nucleotide sequence, is going to bond to the codon. The codon is going to bond to an amino acid residue. A charged tRNA is a tRNA with an amino acid. Okay? This is quaternary structural level. Ribosomes also are quaternary structural. They have a lot of nucleic acids in them. There's a combination between protein and nucleic acids that come together to give you a lot of these enzyme structures and to catalyze reactions.
Is there something like quaternary?
Quaternary?
Quint, like 5, no?
No.
Okay.
Okay, how do we feel? All right. I got a couple more questions for you. There's one.
I would say GNC. Maybe it's the same, I don't know.
All right. Talk to each other.
I did GNC stronger, but I could be no difference.
Yeah, I said GC.
Because of the tree bonds.
Yeah.
So if you want to express a gene, what sequences are you going to look for?
AUG.
To express it, to make a gene. A's and T's, we call that what box?
ATA.
ATA box. Okay?
Tada.
ATA or TATA. Either one.
I like that.
ATA is reverse for TATA. So, biologists are not clever. Life is not clever. So if I look for TATA, the reverse, so this is 5 prime, this is 3 prime, this would be 5, 3 is A, T, A, T, A, T. Atta, tata. Alright? So if this is the gene sequence I'm looking for to express, my gene is this way. If my gene that I'm looking for to express is atta, my gene is that way on the other DNA strand, because they are
Easy.
anti-parallel to each other.
Anti-parallel.
Atta, atta. Now, if I had Gaka and Gaga,
Lady Gaga.
I know, you know what? Since we're having fun, let's just have fun. Scientists are clever. And there's several very funny things that we've done. For instance, scientists like to name things every once in a while that are just stupid, just for fun. One of them decided in the 1980s, I think it was, to name a gene called Sonic the Hedgehog gene. Turns out it happens to be one of the most important genes in regulation. So every paper in the world constantly has to list the Sonic the Hedgehog gene. So it's like the most commonly saying thing. In respiration, which we're going to actually go over this pathway, a researcher decided to call it the fuck yes pathway. Turns out it regulates almost all of respiration. So there's a whole Wikipedia page on funny science names on things, so have at it tonight. With that said, how we're doing the central dogma. We're doing this in pieces. Today and through tomorrow and Wednesday, we're going to do translation. We're starting from the reverse.
So what was the answer?
Because you're most familiar with right now how to build a protein. That's what we've done. Okay? NCC is how you build your protein sequences. So we want to assemble proteins. That is called translation. That is done by what organelle?
Ribosome.
Ribosome. Excellent. So the ribosome, which we just demonstrated, is a protein with what else attached to it?
Ox. Oh, the ribosome?
The ribosome
Oh, with
is a
a-
protein with a
X.
RNA strand. So it can read RNA because RNA can bind to RNA. So it's going to read RNA and use that protein to assemble. Okay, that is the purpose of a ribosome. So this is translation. So we're going to do translation, which is the end process. Then we're going to go to transcription. Transcription is how to make the mRNA to be translated. Then we're going to understand DNA. Once we understand DNA, we know this process. Now we need to start figuring out how to regulate. Well, today you've already learned some regulation. Learned atta, tata, gaka. That's some regulation. You've also learned histination. We can histinate it. We can wrap it up. That's regulation. Okay? So this is the central dogma. The central dogma is hard. All right? So we're going to preface this. The next 3 weeks is hard. It's brutally hard. We are not going to fall behind. We're going to keep up. We're going to keep going. We're going to ask lots of questions. We're going to do this. I'm going to provide you with lots of supplemental information. On Friday, after your exam, I sent out an announcement. And that were 2 books. Okay, we have a bunch of those books laying around here. Renee Fester and myself have written multiple books. A couple of them have literally been on this topic, genetics and cell division. And why those are kind of nice is they're for dummies, which are really cheap books. You can find them for like 50 cents on the Amazon stores, right? A couple of bucks here and there, or we have some. I'm also going to send you out some chapters from a new textbook that's going to be made here in the next couple of years that we're writing right now. It's a companion for this class, so we're updating this class. So use these resources. With that said, additional resources that are decent for this, I'm going to post a whole bunch of stuff on Canvas with links and other stuff, because we want to just be bombarded. Honestly, you spending 4 or 5 hours a day on this is not going to be unreasonable. Okay, it's going to be a lot. Khan Academy is decent. Okay, decent for this. Not all of it is great, but Khan Academy is decent. Some of the stuff we're going to go into is a lot more advanced than they would touch on. So just be mindful of that. Okay, so we're going to do translation, then transcription, DNA, all in the hopes of regulation.
So
This week in lab, we're going to understand transcription and DNA structure. That is our goal. It's a dry lab. It takes a long time. So the lab is a full lab. Okay?
So does it go back and forth from like protein to RNA to DNA, or
Correct.
DNA to?
All the above.
Okay.
That is what makes things hard. All reactions are, all reactions
Reversible.
go either direction. So if I take proteins, I can assemble DNA. But if I have DNA, I can assemble. There you go. You've also now just solved the origins of life. Okay, let's have at it. We're just gonna.
What was the answer?
G and C. This shouldn't be needed. Explain this one.
Well, how do we know? We always get tricked.
The hitting of knowledge is coming. Five is carbon. Carbons. What's that? Yes. It was on campus. It was there when it's not going to do any good now, right?
Yeah, but well, that one is for sure.
This is a ribosome, and it's reading